All right, so we're talking about the nervous system, how it progressed, how it changed through evolution. Nervous system allows or aids in animals of any type to do more complex situations, to respond in a more complex manner to any kind of stimulus. And these stimuli are things that could take away or lessen the chances of survival, like food, like shelter, like reproduction, like stimulus response. So the more we evolved, or the more organisms evolved, the more complex the nervous system actually became. So a little bit like I did last time, just very simplified. We've already been into these very in depth, but this will help with the progression or the changes of the nervous system. Invertebrates' nervous system. Sponges have no nervous system. Cells respond to the environment and release ions, which close and open the osculum. Remember, the osculum was the opening. Ions are charged particles like hydrogen ion, sodium ion, which is Na positive. So the sponges only have these cells that respond to the environment. So there is no true nervous system. Hydras have something called a nerve net. Okay, that is where neurons communicate with one another, and with contractile cells contract and relax. So neurons communicate with one another by chemical signals, and those chemical signals communicate with contractile cells. Contractile, they contract and relax. All right. So that is, now we've gone from sponges to hydras. Notice how the progression has gotten a little more complex. All right, now we move to planarians. Those are flatworms, remember. Cephalization is concentrated or made up mostly with a nerve cord. They're called ganglia. Ganglia are bunches of nerves. They might make a grouping or a ball, if you will. They're sensory receptors, and they send information and react to stimuli. Cephalization means the development of this nervous system. So cephalization has occurred. Again, that's progression from the first two. And then annelids and arthropods, the brain and ganglion are both part of it. And what happens is the brain receives information and controls muscular activity. So it's simplified, but again, it's getting closer and closer towards vertebra. In vertebrate nervous system, there are true divisions. They are more complex as we run through the different types of vertebrates. There are different parts of the brain. You have the hindbrain, which is the back of the brain. You have the midbrain, which is the middle of the brain. And you have the forebrain, which is the front of the brain, and it is significantly larger than the three other parts. Okay? And then the neurons, though, neurons are the primary cells of the nervous system. They are what does the communication. They are what does the sending an impulse. We'll talk more about that in a second. All right, and then the brain itself is divided into two different areas. You have the central nervous system, or the CNS, which includes the brain and spinal cord. Then you have the peripheral nervous system, which represents all the nerves that come off of that central nervous system, off the spinal cord. and it goes in both directions. So the nerves can run from the spinal cord to a particular location of your body, and it also can be, the nerves can run from that particular location of your body back towards the spinal cord. So they go in both directions, not in the same neuron. So a neuron information can only go in one direction. So if we're talking about brain to finger, or spinal cord, excuse me, to finger, there is a nerve that goes from the spinal cord, we'll call it nerve A. Nerve A goes from the spinal cord to the finger, and then nerve B goes from the finger back to the spinal cord. Okay? All right, so let's get into a little bit stuff more in depth. So there really are two categories of cells that are associated with the nervous system. And we'll talk about neurons in a second, but the first grouping are called neuroglia. Neuroglia, if you will, are known as filler cells. They fill up in between the neurons. Okay, I have them listed, and it kind of explains, it kind of tells it what it is, but let me explain it a little bit better. So astrocytes. All right, so neurons need a constant supply of glucose and oxygen. And the only place to receive this constant supply is through a blood vessel, from blood. So an astrocyte literally holds the neuron in place next to a source of sugar or glucose and oxygen. Okay? So it holds it in place. Microglia are cells that monitor what's going on around them. They literally are trying to see, are the cells surrounding them dying? Are they dead? Is there something wrong with them? Or is everything working correctly? And then they can cause a response. I always say, if you have like a line, let's just say it's your charge cord. Your charge cord, you plug it in, it works just fine. But what happens if I cut a piece out in the middle of your charge cord? Then what would happen? Well, that phone would never charge because there's a piece missing. And I want you to think of kind of the nervous system in that manner. Neurons must line up to send this impulse, but what happens if one of those neurons doesn't work? And that's where microglia kinds of pays attention. Ependymal cells are cells that have cilia on them. So think about it this way. There's this fluid that's found surrounding the brain, surrounding the spinal cord. It's called cerebral spinal fluid, or CSF. Cerebral spinal fluid can never be stagnant. It always must stay in movement. It must be in constant movement. And ependymal cells do this movement. It moves that fluid around. It goes back and forth and kind of shifts that cilia. It allows for that motion of cerebral spinal fluid. And I want you to think of it this way. So in Mississippi, you're naked and afraid. You are stuck in a rural area, and you have a choice to drink water. You have a choice from a creek, a portion of the creek that's not moving, and you have a portion of the creek that's swiftly moving. So which one would you drink? Well, I hope you would answer the swift moving area of the creek, because if you were to do it the other way, that stagnant creek is going to hold tons of bacteria growth. And it's the same kind of idea. Your cerebral spinal fluid has to be in motion. Okay, oligodendrocytes, they are going to produce myelin sheath, which I'll talk about in a second, and Schwann cells, which also produce myelin sheath. Now look at this listing that I have of these neuroglia. Do you notice the CNS and PNS? So all the ones that have CNS, those are only found in the central nervous system. The one in the Schwann cells is found in the peripheral nervous system. So the first grouping was neuroglial, those filler cells. The second grouping was going to be neurons. Okay, neurons are the basic or main functioning cell of the nervous system. Okay, and you should remember these structures, but I labeled it just in case. You should know what the structures are and what they kind of do, their functionality. So the cell body is just like any other cell. It has the usual organelles that you're familiar with. Cell body is also called a soma. Okay? Dendrites are projections coming off of the cell body. Dendrites are the receptor region. They receive impulses and they increase the surface area of the cell body. Then we have the axon. That is a single projection coming off of the cell body, and its main goal is to transmit an impulse in one direction. And it goes from cell body towards the axon terminal branches. And then the axon terminal branches, that's at the end of the axon, and their job is to make and release neurotransmitter. And then there's that myelin sheath that I mentioned that is made by oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system. And what they are, myelin sheath is protein lipid mix that wraps around the axon. And if myelin sheath is present, the strength of an action potential, and I know you don't know what action potential is yet, but just write down the strength of an action potential is stronger and the speed of that impulse going through the axon is faster. And I'll use that same type of example of the cell charge cord. So if you look at a charge cord, you notice there's a plastic coating over the charge cord. So if I peeled it back, you would see that there is a metal strip, if you will. So I want you to think of that plastic coating as the myelin sheath. So if the myelin sheath is there, it's intact, you plug the cord in, providing there's no other damage, your cell phone charges pretty quick. It's efficient. Okay, so what happens if that plastic coating is peeled away? If it's peeled away, then did you notice that then the charging rate lessens? It gets less quickly. It's less effective. You got to bend it a certain way to get it to work. So think of that analogy when you're understanding what a myelin sheath does to an impulse. So when we talk about types of neurons, okay, there are different types, categories, if you will, of neurons. So the first one is a sensory neuron. A sensory neuron sends information towards the central nervous system. Okay? Sensory neuron sends impulses towards the central nervous system, so therefore it is afferent. An interneuron is found in the central nervous system and it directs traffic. It directs traffic. What it means is when an impulse comes to the central nervous, that interneuron now is going to send it where it needs to go in the brain. Okay? Then you have your motor neuron. Those are impulses that come away from the central nervous system and they cause movement. And if they're moving away from the central nervous system, they're efferent. Remember, E for exit. They're efferent. There's something called a reflex arc. So a reflex arc, okay, a reflex arc. So reflex, you know what a reflex is? I punch you, you cry. No, I punch you, you turn around and try to punch me back. That's a reflex. I hit your knee, what happens? Your leg jumps. That's a reflex. So there's something called a reflex arc, which means there's a clear pattern to what happens. So you've got a stimulus. So you've got a stimulus. I'm just going to put S. Stimulus like fire. Then you have a sensory neuron. I'm going to just put SN. The sensory neuron now goes towards the central nervous system to the interneuron. Okay, the interneuron is going to direct traffic. Then it goes to the motor neuron, right? And lastly, to an effector, something that actually responds to that stimulus. That is the effector. Okay? So now we're getting into the more complex, remember, something you can't touch, you've never dealt with. So try to think abstractly as best you can. So either information or response to send information is what a true nerve impulse is. A nerve impulse sends information or sends a response to a stimulus. The purpose of a neuron is a way to send and receive information all over your body. And as long, remember, we're always talking about people with no signs or symptoms or disorders, they're quote unquote normal. Then normally it is specific. It's almost predictable. And you can actually measure an impulse. You can actually measure, if you will, the voltage of an impulse. It would be like if you had this axon that goes in this direction. If I probe right here, so I stick a measurable voltmeter right there, and I stuck a voltmeter right here, I can measure the voltage in that area. It's a voltmeter, and it has the two electrodes, and it does tell a charge. Okay, so here comes that weird stuff that, the stuff that is really hard to understand, but I want you to bear with me. So remember, if we take, okay, now remember my drawings stink. So there's your cell body, right? There's the dendrites coming off the cell body, right? And there's that axon. And an axon has membranes, almost like a cell. See that? That's the axon. Now let's blow up. Now remember, this is microscopic. I can see under 40x very easily a neuron. Let's blow it up so it's life size. So I'm blowing that up. All right, so let's blow it up. Let's look at it in a size that will sort of make sense to you. So I'm blowing it up, right? We can agree this is the same thing except for much bigger, right? We can agree that an axon has two membranes, okay? And you can see on either side there's an external environment surrounding the axon and internal environment. So this is internal, this is external, right? I'm going to just I'm going to put an E right here. Just so you're aware of where we're talking about. All right, so there's something called a resting state. A resting state means that nothing is being transmitted. There's no impulse going through this axon, but it is ready for an impulse to come. It's at rest, but it's ready, right? So if we talk about what's found on the inside and outside, so on the inside, the internal environment, you have higher levels of potassium. Potassium is K positive, right? On the external environment, or outside the axon, you will have high levels of sodium. Which is Na positive, right? And there is a charge inside. Remember I said I can measure the charge anywhere with the voltmeter. There is a charge to a resting membrane, right? This is at rest. You know there's membranes because it separates internal from external. The charge is -70. Okay? This means if you drew that for me or you look at it, you would say, okay, nothing's happening, but it's ready to roll. And I want you to know something, and I want you to never forget this when we're dealing with anything in an organism's body. If we change the charge, something will happen. So, let me go further with that. If we change the charge, especially if we go less negative, so closer to zero, something's going to happen, and I'll talk about in a second. But I need you to remember this. I need you to be able to draw it. I need you to be able to picture it if I ask questions about it. That's resting state. That means no impulse is being transmitted. However, it's ready for an impulse to be transmitted. So how does this kind of work? First, let me define that word action potential. Action potential is also abbreviated AP. An action potential is an electrical charge right, that when the charge changes, depolarization can exist. It's like an electrical current. Okay, so you're just standing there and I want you to move. Well, you're just standing there, and if I say move, you're not doing anything, right? You have your earbuds in, you can't hear me, don't move. However, if I take a cattle prod and pop you, you're going to move. Think of that transmission of the electricity from a cattle prod as being an action potential. Action potentials go in one direction down the axon, from cell body or soma to the terminal branches. So you can see that on this picture here. It's going to go in that direction only. It's not back and forth. Like, you're not going to send a transmission in this direction. You know, it just does not go that way. It only goes one way. So that doesn't happen. Okay? Okay, so when we talk about how it works, or what it does, is the action potential must travel. And remember I said we need to change the charge. So remember what that charge was on the resting potential? Go ahead. Right? -70. So if we change the charge from -70 to negative 55. Negative 55 is a big, big explanation. Negative 55, when that occurs, the action potential is made and it'll travel. That negative 55 is called threshold. Okay, so how do we do that? Well, remember the resting potential, right? When it was at rest in negative 70, you had potassium on the inside, which is a positive ion, and we have sodium on the inside, which is also, or on the outside, excuse me, a positive ion. If I want the inside to become less negative, we want to put positive ions in there, right? If I add positive 1 to this, it's now negative 69. If I add positive 2 to it, it's negative 68. Does that make sense? All right, so if I want the charge on the inside, that resting membrane to happen, I have to open a gate or a channel and allow sodium ions in until it reaches negative 55. Okay, does that make sense? Now, how does it work? So movement from direction to other, it has to change charge. And I put this Christmas tree or these Christmas lights for a reason. So think about it. It has to move. It's not like an all or nothing thing, meaning that all of it goes and changes to negative 55 and races through. It's complicated, and I know I don't want to go too much over your head, but let's just say here's that axon, right? So you have that negative 55 go right here, right? It turns negative 55 here. So the action potential can go to right there. Then all of a sudden the charge here changed to negative 55. Now that action potential can move to here. So it's done in a way of Christmas lights. You know those Christmas lights that looks like it starts and it travels, and when it turns on, then it eventually turns off? So every time that light turns on, that's an action potential, and it travels down that Christmas light series. Remember that. Now, we have to do something from behind. So let me just erase that little star there. Okay, so the action potential is now here, but what if we want to send another action potential right behind it? Well, we have to get it back to resting state. That's called repolarization. So back here where the action potential already started, that's repolarization. So to move, it's depolarization, and immediately where it came from is repolarization. That means repolarization, it's bringing it back to resting state. Again, there's that Christmas light scenario. When the light turns on, it's depolarization. When the light turns off, it's repolarization. Repolarization means going back from that negative 55 back towards negative 70. Well, what does that mean? We have to make it more negative. And we got all these sodium in here with the potassium. Well, if we close this gate and don't let any more sodium in, but we open a gate and let potassium out, remember potassium's positive well, do you see how it would go back towards negative 70? I'll talk about this more, but I want you to play with these drawings and that Christmas light scenario, and you'll get it, okay? Okay, so how it continues. So we just talked about how an impulse, an action potential travels. Again, you know my drawings stink. There's the cell body and the dendrites, how it travels down the axon to the terminal branches, right? It's going in that direction. Well, we need it to actually go to another neuron, because it's not one neuron that goes from your brain to your tips of your toes. That would be ridiculous. It's microscopic. But it has to travel from one neuron to the next, right? And there's a space between the neurons. That's called a synapse. So I'm going to put the space right here as a synapse. That's the space between neurons. And you can see it clearly right there in that blue picture. Those are two neurons. You see the space between them? That's a synapse. So somehow I have to send the impulse from this first one. We'll call that pre, because it's before the synapse. And we'll call this second one post. Okay? So it's going still in the same direction. So the neuron before the synapse is the presynaptic neuron, which is right here written. Then you've got the synapse, and then you have the postsynaptic neuron, which is the one post. It's the next one. And it can go on and on and on as a series to go to the location. Well, there has to be a way for that action potential to continue, not just stop. So remember what I said about those axon terminals. They make a neurotransmitter which helps send the impulse. And there's steps for how this occurs. So I took this image and blew it up. The list of what the steps are, and I know how much you love steps, is a process. What I do is I have a blank slide, and on this next blank slide, I'm going to actually draw it in my own fashion, the steps for making it move from one to another. So I'm talking about we're at the end of the terminal branches and going to the dendrite of the next. See how it's back and forth? So this is going to be the terminal branches, and this is going to be the dendrites. I'm going to blow that up and tell you what exactly happens chemically for that transmission to occur. Because we've already talked about it going down the axon. Now it has to jump, if you will, from one neuron to the next. Here is that blank screen like I promised. So go ahead, and I want you to draw what I'm saying and label what I'm saying. It will make sense because you can read the other one. Reading it's one way to memorizing it, but actually writing down the steps will make sense to you in a drawing. So if you will, you know my drawings stink. So this is the terminal branches of one, and I'm just going to put terminal. Okay? This is the dendrites of the second neuron. Right? Okay. This right here is the synapse. We're good so far? Terminal branch of one neuron, the presynaptic neuron, is in blue. The yellow is the dendrites of the next neuron, which is the postsynaptic neuron, and then the synapses between the two. All right? So I'm going to do the steps in white. Okay? Step 1. I'll do the steps in different ones. Step 1. The action potential arrives. Remember, the action potential is traveled down that axon and reaches the terminal branches. So I'm just going to put action potential. Okay? And I want you to think of when you send an electrical current, doesn't it have reactions? Okay? It has like a domino effect reactions. So the action potential arrives. That's step 1. And I'll just put a 1. Try to keep doing different colors. Okay? Step 2 is because of that action potential, calcium gates open. So I'm at the terminal branch still. Gates open. A gate is a channel that's normally closed, but because something stimulates it, it opens. And I'm going to do the chemical symbol for calcium is Ca, just in case you didn't know. And it's a Ca, and you know how normally sodium is a positive 1? Calcium is a positive 2, so I'm going to just put plus, 2 pluses here. Calcium gates open, and calcium floods in. Now, do you notice that the charge will change? The charge that's in that terminal branch is one thing, and now all of a sudden I've got all these positives coming in. So we agree that the charge is going to cause something to happen, like it always does. Then what happens is, because that calcium is flooding in, remember I said, what does the terminal branches produce? That's right, neurotransmitter. So because that calcium is coming in, neurotransmitter is going to be put in a vesicle. I'm going to put a little N in here. And it's not just one, there'll be a bunch of them, but I'm just going to draw one. Because of step 2, neurotransmitter is put in a vesicle. What's a vesicle? A vesicle is a bubble. And think about it, it's almost like when you take, if you just took an aspirin and swallow it, you can taste it, right? And it's mostly dissolved by the time it reaches the stomach. But what happens if you take a time-released, a capsule of Tylenol? That capsule protects the medicine on the inside, so it doesn't release before it should inside the stomach. Same idea. If the neurotransmitter is just released, well, it would break apart. It would have a chemical reaction and wouldn't be as strong as when it gets to the other one. But if it's placed in this bubble, it's placed in this vesicle, nothing's going to happen until it's released. Got it? Okay, so let's move on to step 4. Step 4, let me do a different color. Let's do yellow. Step 4 is that neurotransmitter travels down the axon terminal and released by exocytosis into the synapse. Again, the neurotransmitter in that vesicle travels down to the very end of that terminal branches, and it's released by exocytosis. And where is it now? That's right, it's in the synapse. Okay, step 5, another color. Running out of colors here. All right, we'll do this. Step 5, that neurotransmitter crosses the synapse and binds to receptors found on that postsynaptic neuron. So there it is. There's the receptors, and here it's binding. See them binding right there? So step 5 again. The neurotransmitter crosses the synapse and binds to receptors on the postsynaptic neuron. Step 6 is real simple. The action potential goes on. So now it's at the second neuron, and then now the impulse can travel. Draw this, label it. Reading it is one thing, and your brain doesn't like all these paragraphs. It can't organize it as well as if it were to remember it by a picture. So this works. Okay? So let's talk about the divisions, and I'm not going to go into different things, but let's talk about the central nervous system. The central nervous system is basically the control center, and remember, it includes the brain and spinal cord. This is the center that actually receives impulses. It processes what you need to do and sends a response. Okay, so let's start with the spinal cord, which that is a real human brain. On both pictures, those are cadaver dissections, so you can clearly see what it really looks like. The spinal cord receives impulses and it sends it to the brain. It also receives information or impulses from the brain and sends it to the parts of your body or the regions of the body that needs to react. It's made up of the same exact tissue as the brain. So if the brain has gray matter and white matter, it also has gray matter and white matter. Okay? So let's see. It's branching, which you can see in this image over here. You can see there's tons of nerves coming off or going towards the spinal cord. You also can see it's highly sensitive. It's not only protected, which you say, by bone, which is the vertebral column, but it's also protected by 3 layers of membranes. They're called meninges. And these 3 layers of protection, these meninges, are all different types. And I can honestly tell you, if you were to do a dissection of the brain also has these meninges as well, this protective coating. These meninges, if you touch the outer meninge, it's called the dura mater. It literally feels like plastic. It's very hard. So there's these layers of protection to try to ensure no damage of the spinal column. Now, referring to those 2 types of tissue, it's very easy. Remember I said myelin sheath is a protein lipid? It actually is creamy white. So white matter, therefore, would be made up of myelinated fibers. It has that myelin sheath. Gray matter, it's darker in color, and you can kind of see in that depiction, that cartoon depiction in the center, gray matter is made of unmyelinated fibers and cell bodies. So the brain itself, okay, so extremely complex. Every nook and cranny, if you will, has a specific job and actually has a name, and you're really thankful that you don't have to remember all these because we're not in gross anatomy. But you can see there's where I talked about that meninges. See that bottom left-hand picture? There's that dura mater. You can look at it. That's exactly what it looks like, okay? Every bump is called a gyrus. So see that bump right there? So every bump is called a gyrus. Every little dip, if you will, is called a sulcus. So there's a dip right there. Okay? Every big dip, every big clear separation is called a fissure. There's that longitudinal fissure that separates the right and left hemisphere. And you can see I've opened it right here. And then you also have a fissure here. See that separation between the cerebrum and cerebellum? So there's 2 great fissures. Longitudinal means it runs the length. Transverse fissure means it goes side to side, and that separates cerebrum and cerebellum. There's 2 hemispheres. There's the right hemisphere and the left hemisphere. Cerebrum, cerebellum, gyrus is the bump, sulcus is the little dip. Remember, there's the meninges that protect the cerebrum, protect the brain and spinal cord. And remember, CSF, there has to be fluid, because if there any portion of the brain or spinal cord desiccates or dries out, an impulse can't travel. Remember those ions that I talked about around surrounding or inside the inside and surrounding the axon? Those ions are present in cerebral spinal fluid. No fluid, dries out, no ions are present. There's no way you can go from resting state to depolarization. Impulse doesn't travel. Just some basic structures internally. I'm not getting into everything. I put little arrows to help you find it. First thing when I talk about it, do you see a bird? No, I'm not crazy. Do you see a bird? I clearly see a bird. There's the head of the bird, right? There's the body of the bird. There's a headdress. The only thing you don't see is wings. Okay, so keep that image alive. So if we talk about it, I'll go from the bottom to the top. And let me use a darker color to kind of help you out. There's your spinal cord right there. This first little bump is going to be the medulla. The big bump, which I call the bird's pregnant belly, is going to be pons. The medulla, while I'm here, the medulla is involved with the heart, beats, and breathing. Pons is involved with respiration, swallowing, bladder control. This little berry, if you will, that the bird is holding is the pituitary gland. I'm just going to put pit. The pituitary gland produces 6 hormones and releases them. That's as far as I want to go right now. Then that eye of the brain, of the bird, see that eye right there? That's the thalamus. The thalamus is the receiving and sending information. It's like a post office. It receives mail and sends out mail. Then we go to the back here, and there's a little tiny gland right there. That little tiny gland is called the pineal gland. I'm just going to put pine for now. The pineal gland is involved with your sleep-wake cycle. Okay. See the stuff right here in cerebellum? It looks like trees. You can actually see it's realistic in this real, that's a real dissected brain. You can see it looks like the trees. That's called arbor vitae. That's this right there. Arbor vitae means tree of life. And it does hand-eye coordination. It does those really complex controlled movements like picking up a pen. Okay, a couple other things, and I don't have it on the list. Right here in this location, inside the beak, this location, that's called the hypothalamus. Let's put hypo. The hypothalamus regulates thirst, hunger, and temperature. Okay, so layers of the brain, very simple. I'm not getting into all of it. Obviously, there's protection of the brain. So you've got not only have the meninges, you have the cerebral spinal fluid, you have the skull. So there's several layers of protection. Okay, tissue is there. Now I did here and here. So you can clearly see white matter, gray matter. So gray matter is on the outside. That's called the cortex. So that's the outermost. White matter is the next layer in. And then basal nuclei is that deepest layer. Okay? Made up of all we want. The cortex, the cerebral cortex, is the outermost layer of gray matter. It's called the cerebral cortex. Cortex does almost everything. It's involved with learning. It's involved with memory. It's involved with language, with speech, with senses. Okay? And again, I'm not getting into as heavily as I would for A and P, but there's several systems within the regions of the brain that play a role in everyday life. It's a checks and balances system. There's always primary and associated areas. What I mean by primary associated areas, let's just take, and they're all involved with sensing. So if let's just take, you have a primary visual cortex. Primary visual cortex is your primary sight. You know you're looking at something. Then you have a secondary area. So you have the primary visual cortex, and I'm just going to put PVC. That area PVC, the primary visual cortex, knows you're looking at something. The associated visual cortex gives you a fine, detailed description of what you're seeing. Down the hall, you see what looks like a boy. Well, the associated visual cortex would say, "Oh, I've seen that boy before. That's Carson." Okay. The limbic system gives emotions to an area of the brain. So, you know, when I see this person, I smile because I have affection for them. Or when I see this person, I get really angry because I hate the person. So that's the limbic system. RAS stands for Reticular Activating System. RAS has nuclei and nerve fibers, and it runs between the spinal cord and brain in both directions. It's almost like a connection between the brain and spinal cord. RAS is really important because it turns on cerebrum. It makes a person alert. It's kind of like you're sleeping, the alarm clock's going off, all of a sudden you wake up. That's RAS. Lastly, I'm not going to say this, but I've organized everything for you to understand how the nervous system is organized.